My name is Mike Winters. I'm a PhD candidate in music technology at the Georgia Tech Center for Music Technology. And I'm in the Brain Music Lab with Grace Leslie. A lot of my research has to do with the effects of music on the brain. And today is about heartbeats, which I think by the end of this talk, you're going to have a sense of how those are really important for understanding music listening. The title of my talk is Empathic Effects of Auditory Heartbeats, a Neurophysiological Investigation. And so we're going to be looking at the ways that the heart is able to modify a person's empathic state, and then what happens to their heart and their brain when this happens to them. So let's just, this is the heartbeat sound. Oops. One sec. Good. Okay. So I'm assuming many of you are enjoy music and listen to music. I've noticed a few things though. If you listen to lyrical music, the heart is a very common metaphor. You hear it in lyrics all the time. What does it mean? It usually means something to do with feeling. I did some prior work and I found the top 25 songs with heart in the title, but these are really great. Closer to the Heart, Heart of Gold, Heartbreaker, artists like Tom Petty, Rod Stewart, the Beatles. The heart is everywhere. But if you also listen to music, I think you'll find the heart is there too. Let's listen. The musical beat in particular, the tactus, and the heartbeat are very similar. Let's listen to some music that sounds like heartbeats. Okay, and so when you listen to music in this way, you can kind of see how the heartbeat is everywhere. The heartbeat is very similar to the beat of music. The final thing that I hope you all realize is that music is a very social and shared thing. When we go to concerts, when we listen to music, it's often in a social context. Performers, composers are often performing and composing for other people. There's a desire to share this information. Common music apps like Spotify make this really easy. They have a share button. That's for you to share your music. So my motivating questions behind this research are, why is the heart such a prominent metaphor for feeling? Why does the beat of music resemble a heartbeat? And when do we feel with music? I don't have definitive answers to this, but I think by the end I'm going to have some pretty good answers. This is the outline of my talk. I'm going to start off with giving you a sense of the background behind what I'm doing. Then I'm going to describe an experiment that allows me to test for the effects of heartbeats on other people. And I'm going to present three kinds of results: behavioral results, ECG results, which are how the heart is responding, and EEG results. Then I'm going to follow it up with a discussion that is going to bring all these results back into significance for music. Let's start off with the background. Physiological signal sharing is probably something you haven't heard of before, but it's happening. All over, there are wearable physiological sensors. This is the Apple Watch, and it comes with a device for measuring your heart rate. There's also context of remote communication. The people that we love, the people that we care about are often physically removed from us. We want to communicate with them. We also have a desire for intimacy and closeness, to connect with those people and to share our feelings with them. In this context, heart rate sharing applications have arisen in the context of HCI. People have been trying to understand what exactly happens when we share our heart with someone else. They've identified two axes or two fundamental characteristics of this signal sharing process. One is the heart rate as information, the heart rate telling you something about the other person. So for this example, someone texts their heart rate and they say, what's the situation? There's some information there. Then there's this heart rate as connection. And I think that's kind of what's behind the Apple Watch. This is the digital touch app. And if you have a an Apple Watch, if you hold 2 fingers down, it will collect your pulse and then create a visualization that beats at the beat of your own heart. And you send that out. But anyways, researchers have identified this idea, this desire for heart rate as connection. What's really surprising about these apps, though, is that they're visual, whereas common experiences of the heart are tactile, like when we take our pulse, or auditory when we listen to someone else's heart. And so this is a really important question for me as a musician. What are the effects of the auditory heartbeat? And in particular, can heart rate sharing be an effective empathic technology? And if it can, then we have to go into the science of empathy. So what is empathy? If you haven't heard this term before, it's the ability to understand and share the feelings of another from their perspective. There's 2 types. The first type is cognitive, which is the perspective taking of another person, the ability to identify that person's emotion. I can tell you're angry, or I can tell you're sad. Then there's this affective component, which has a lot to do with your ability to share in those feelings. A great example of that is emotional contagion. And the affective component has to do with your own feelings. There are behavioral correlates of empathy, which allow us to study it from a sort of scientific lens. This includes things like mimicry, entrainment, contagion. And then there's also neurophysiological correlates, which are things that we can measure from the body, like EEG, electrocardiogram, EDA, which is your skin response. And an important question for me is whether music can alter this empathy, or music can affect this connection between people. Well, the good thing about music is that it's already very, very social. Group musicking is, music as a verb is musicking. And musicking often occurs in groups. When it does, it has this capacity to synchronize bodies and moods. Synchronizing bodies is dance, moods is emotions. It has the capacity to increase affiliation, cohesion, and teamwork. It's also supported by neurochemistry. That when we listen to music, we actually undergo neurochemical changes that relax us, that make us more prone and more pro-social, more able to get close to other people, less aggressive. Also, in studies with children, musical group interaction in schools has been shown to actually increase trait empathy in people. And some people have pointed, in fact, to this group capacity as an evolutionary function of music. People ask, why do we have music? Is there some sort of role this has in our culture for keeping humans around? And this is a very promising explanation for that, this group effect. There's also empathy in music listening. Empathic traits have been shown to predict preferences for music, predict preferences for genres, preferences for particular types of music, fast or slow. It also predicts the ability to synchronize. People with higher empathic perspective taking tend to be better at doing musical coordination tasks. Finally, music is embodied expression. Until the 20th century, when we heard music, it was very embodied. You have to make music, you have to actually you have to play an instrument. And also this idea of expression, which is so fundamental to music, the idea of music expression. I think that term itself is very tied up with another person. Music is expressing something from one person to another. And this has to do with embodied music cognition. Also, it's listening to music, we can empathize directly with the performer that's performing. I think that's what's happening when we listen to our favorite artist. We can also empathize with the composer. And finally, there's a very interesting possibility, which is that music can actually be a virtual persona. That is to say, we can listen to music just the structural and acoustic content as if it's another person. And this is a really interesting idea. People have pointed to the beat of music as being indicative of walking or someone moving their body. Melodies might be more like someone singing or someone talking. So in this way, music can be an embodied, expressive, virtual persona. All of these things, though, are external. We're imagining the body of another person, how they're moving. And for my research, I'm interested in whether we can also empathize with internal physiological states, things that we can't see, things that are happening inside the body of another person. This is actually something that's been going on for quite a while in music. This is Alvin Lucier. He's playing a piece called Music for a Solo Performer. And this is generating EEG from his brain that's being used to generate music. And this is something that happened in the 60s, roughly as early as we could be making music from biosignals, we were doing that. These days, though, people are exploring biomusic as an empathic or a communication technology. So this is actually work with people with autism, and they've mapped electrodermal activity to melodic pitch, skin temperature to key change, blood volume pulse to tempo, respiration to a whooshing sound. And this is just one of many possible mappings of internal physiological states into music. And for my research, what's really important to me is this question of, will listening to a person's physiology affect our physiology? So just like when we see someone dancing, when we start to dance too, when we hear someone's heart beating, does our heart kind of have a response to that beating heart as well? If it does, it probably interacts with our autonomic nervous system, which is roughly regulating and deciding our physiological state. Music is full of structural cues that have the capacity to induce emotions in us. And one of the strongest of those is the beat. The beat can interact with empathic mechanisms, things that are outside of music, which are fundamental to our social capacities. And people have identified two particular mechanisms in particular that are, in my view, empathic by nature. Those are emotional contagion, which outside of music is our spontaneous mimicking of other people's bodies. So when you see someone smile, you smile. And then rhythmic entrainment, which is the spontaneous, the way that our body spontaneously seems to entrain to music. And so both of these have the capacity of generating induced emotions in us. And when there are induced emotions, there are physiological changes. So this is the route that an internal physiological state could take to a physiological change in the listener. When these two interact in my model, the result is an affective shift. So the affective shift can take two kinds of ways. One is it changes the perspective or the recognized emotion in the listener. So I now think instead that you're not angry, but you're probably sad. And then this can also create felt emotions in us. And so these are all part of the affective shift. And for my research, the fundamental question is, can just one musical variable, tempo, create this change? So now getting to the research. Does hearing the heartbeat of another person increase listeners' empathy with that person? And does it change their cardiac physiology? Let's get to the experiment description. I have three hypotheses. Listening to the heartbeat of another person will change empathic state. It will change the listener's heart rate, and also cardiac cortical processing. This has to do with your brain's own processing of your afferent cardiac signal. And so the three results, the behavioral results, the ECG results, the EEG results will all reflect these hypotheses. So now to do my experiment, I need to create a heartbeat sound. And I do so using a computer music language called Super Collider. This gives me a lot of control over what the heart sounds like. So for me, what the most important thing is the ability to control tempo. I want to be able to create slow heartbeats or fast heartbeats or anything in between. It also allows me to just create some variability in tempo and loudness. This is for increased realism. And it all starts off with just one heartbeat sample. Okay, and that's what it sounds like. Okay. There is, the heartbeat is, some people have asked me how I chose this sound or why I chose the heartbeat. And the answer is because it sounds like a musical beat to me, and because of these similarities. And in terms of how I chose this particular sample, I went online and I searched all of the different heartbeat sounds. I chose the one that I thought sounded most like a heartbeat to me. But what a heartbeat sounds like is completely dependent upon the context, whether you're a doctor, whether you're sonifying an electrocardiogram signal, whether you're at the movies, whether you're in a video game, the heart is going to sound very different. Okay, so from this, I can create a slow heartbeat sound. This is a very slow heartbeat, 40 BPM. Fast heartbeat sound. And we can also create like arbitrary modulations. And see what's happening to a listener as that's happening. Bruce, did you have a question?
Could you help us understand why you consider 40 to be slow and 120 to be fast?
Yeah, so 40 BPM would be probably the slowest musical tempo that you will encounter. This is and 120 is you can go faster than that, obviously, but that's a pretty good fast tempo, and you want to know.
Derived from heartbeats?
Yeah, they are. So there's only 2 tempi here, but it's roughly within the range of normal human heartbeats. It's way outside of like the normal range. So I mean, 120 would be common if you're working out, or if you just have a high heartbeat signal, and 40 would be you're in incredible shape or you're sleeping. So anyways, the spread is actually to create a very large contrast between these two, which allowed me to kind of very clearly distinguish the effects of slow and fast heartbeats. Okay. To measure empathic state, this is I'm using something called the Reading the Mind in the Eyes task. This is a data set of 36 sets of eyes. And each set of eyes comes with a question, which is, what is this person feeling? And in this test, in the normal version, you have to choose one of these 4 words. There's 4 words in each of the corners, and you just have to choose one of these. There's not a correct answer, except for what people generally agree is the right answer. But for my work, I actually take these stimuli and I modify them by adding a heartbeat sound. So this person, for example, this is a fast heartbeat sound. And I'm curious if hearing this heartbeat changes your connection to this person, your empathy with this person. You can also hear a slow heartbeat. And so maybe this person seemed annoyed earlier, but now they seem preoccupied with the heartbeat. I take 2 responses for each set of eyes. One is their cognitive empathy, which is a simple question of what they are feeling, what is this person feeling? And then affective empathy, which is how well you felt what they're feeling. This is a participant's response. And the reason I chose this is actually because it's a very, very common instrument for studying social cognition in general. It's been shown to be indicative of someone's or of autism, for example, and helpful in the diagnosis of that. Also because the eyes are very, very expressive. And so we could have all sorts of other things like gestures and faces and all sorts of stuff, but just the eyes, I think, gives me a lot of control. And there's 36 of them, and because this is such a commonly used instrument, I went with this one. And so in my experiment, I have 4 basic conditions. Auditory only, visual only, audio visual slow, audio visual fast. And each of those presentations is the participant has to answer a question, which is, what is this person feeling? that stimulus exposure lasts for 20 seconds, and then there's 10 seconds where they answer the second question. How well did you feel what they're feeling? And this period of 10 seconds is a baseline, it's silent. And I use that also to baseline the subsequent trial, which is to have their heart rate return to normal, have their physiology return to normal. I randomize these conditions. So basically, there's no ordering. Every participant has a different ordering. But in general, the 36 eyes happen once, twice, 3, 4 times in a row. So there's no like, there's some spacing in between subsequent presentations. And then yeah, the audiovisual, slow audiovisual, the 4 conditions are then dispersed evenly between those. Let's see what it was like for participants. This is the audio only condition. And this is the affective empathy question. This is the visual only condition, no heartbeat. This is audiovisual fast. And audiovisual slow. Okay, and I did not map the eyes into sound, so I did not say these sorts of eyes are, we'll go with these heartbeats and these other eyes will go with these heartbeats. So, so to accommodate for the fact that sometimes the eyes will not match the heartbeat, I have an additional variable called audiovisual congruency. Yes?
Does this data set of the eyes actually have an answer, correct answer?
Yeah, it does.
So you see that?
Yeah, so there is a correct,
yeah.
There's a correct answer, and you can kind of see it. It's lightly bolded here, but each of these has the correct answer. And when you use it for diagnostic purposes, you take into account how many they got correct. But because I'm modifying this, I'm no longer looking at correct or incorrect responses.
That was my real question.
Yeah.
Do you have a correct answer once you modify it?
I think that there's not a correct answer anymore. But so what I do instead is I just look at change. Whether there's a difference between the visual only condition versus the audiovisual.
You may want to just be careful when using the word correct.
Yeah.
Yeah. I don't like to use it.
Yeah. It's population stereotype, which
Yeah.
is the most common.
Yeah.
Sort of the modal response.
Yeah, exactly.
Yeah, they have been validated too, so.
We're intending to display some sort of particular emotion, so
Yeah.
whether they did it correctly or not.
Yeah, this is a validated data set. So to determine whether the eyes match the heartbeat, I then sort these answers by their arousal. This is some separate research that has looked at the emotional content of individual words, and I've sorted these now by the arousal mean here. So these are low arousal words, and these are high arousal words. Now, in the low arousal group, a slow heartbeat is congruent, and a fast heartbeat is incongruent. And in the high arousal group, a slow heartbeat is incongruent, and a fast heartbeat is congruent. And incongruency, I do not look at this like middle space in between. And so the experimental conditions are something like this. From these 4 fundamental conditions, there are 3 perceptual cognitive variables. The modality, which is whether it was visual only, audio only, or audio visual. The tempo, whether it was slow or fast. And the congruency, congruent or incongruent. This is my technical setup. I start off with a Brain Vision ActiChamp amplifier. This is used for recording 64-channel EEG. This is for the cardiac cortical processing. I also have this is a heart rate monitor, which is for ECG, and there's 3 leads. You can kind of see that here. And then there's an audio interface, which is sending the heartbeats to the listener, That's also being synchronized right here going into the amplifier. So all three of these signals are synchronized that way. I also have some software. I'm using Brain Vision Recorder to get the EEG and ECG data. And then my experiment code was written in Super Collider, and I'm using something called OSC to send markers to something else, which is lab streaming layer, which is then synchronizing the physiological signals with the participants' responses to the experiment. This is the participants I had. There were 6 female and 21 male. 3 black, 11 Asian, 13 white. They were all fluent English speakers. Their age was 19.8 plus or minus 1.2 years. And they had they had about 4.0 to plus or minus 3.6 years of formal music training. This is I'm not sure if this is typical, might be representative of Georgia Tech in general. These participants were recruited from the SONA subject pool. I have a very kind of advanced, I also do some more analysis of their music, but this is just one variable of their musical training. So let's get to the results. So the first question is whether their cognitive empathy changed. So they have this question, what is this person feeling? And they have to make a response. So my dependent variable in this case is something called a change in RMET emotion selection. RMET is reading the mind in the eyes task. And this is a simple change or no change. Did they change their response or not? This is compared to a pretrial baseline, which is done before the experiment even begins in the typical way as you would normally. And so here's the first result of the modality. This is a comparison of the visual only versus the audio-visual condition versus the RMET change variable. And there was a significant effect. Hearing someone's heartbeat changed the listener's perspective on that person's affect. So there was more change in the audio-visual condition than the visual-only condition. The visual-only condition you might expect to be 0, but we expected that there would be changes due to the experiment itself, the interspersed nature, the fact that these trials are 20 seconds, a lot of differences from the normal baseline study. So this would be baseline change, and everything else beyond that is due to the heartbeat. There were additional congruency effects. This is a comparison of the visual only, the audio-visual congruent, and the audio-visual incongruent conditions, looking at the RMET change variable. And from this, we can see that the audio-visual incongruent was significantly higher than the audio-visual congruent. And what's more, the audio-visual congruent and the visual-only condition were not statistically different. This means that when the heartbeat did not match the eyes, listeners changed their affective perspective. Okay. There was also tempo effects. This is a comparison of the visual only, the audio-visual slow, the audio-visual fast. And when I'm saying audio-visual, it means we see the eyes and we hear the heartbeat. And so audio-visual slow means slow heartbeat, audio-visual fast, fast heartbeat. And there was also a significant effect here. In particular, the audio-visual fast changed perspective significantly more than the audio-visual slow condition. And what's more, the audio-visual fast, or the audio-visual slow, and the visual only were not statistically significantly different. So this means that fast heartbeats drive the change in perspective. Let's go now to the affective empathy component. For every set of eyes, for every heartbeat, the person had to answer this question: how well did you feel what they were feeling? We have a dependent variable here called feeling what they're feeling. It's a self-report. It's a 7-point scale. And then to remove bias, participant bias, they might just seem to always have a stronger response. I apply a z-score to their data, which gives everyone the same mean and standard deviation. So let's look at the effects of modality. This is a plot of the visual only, the visual only, audio only, and audio visual stimulus conditions. And this is the feeling strength z-score on the left here. And there was a significant effect. Namely, the audiovisual condition was significantly higher, had produced significantly higher ratings of feeling what they are feeling than either the visual only or the audio-only condition. Something that's really interesting about this is that the visual-only and audio-only conditions were not significantly different. That means that whether you see someone's eyes or hear their heartbeat without seeing their eyes, produce similar levels of feeling what they were feeling. This is something actually that's been found in other research too. But from this, we get the fact that hearing someone's heartbeat increases listeners' ratings of feeling what they were feeling. Let's look at the congruency effects. So this is a comparison of the visual-only, audio-only, audio-visual incongruent, and audio-visual congruent effects. And we can see that there was a significant effect. In particular, the audio-visual congruent produced the highest ratings of feeling what they were feeling. This was significantly higher than the audio-visual incongruent condition. And so this means that heartbeats whose tempo matches the visual affect in the eyes produce the highest ratings. So this would speak to the importance of having an accurate reading of the person's heart rate when you do a heart rate sharing, an audiovisual heart rate sharing. And also let's look at tempo effects. This is a comparison of the visual only, audio only slow, audio only fast, audio visual slow, and audio visual fast conditions. And there's no significant difference between the audio visual slow and audio visual fast conditions. So there's no effective tempo there. But in the audio only condition, when we do not see their eyes, the tempo mattered a lot. When the tempo was fast, when the heartbeat was fast, this produced significantly higher ratings of feeling what they were feeling. So in the context of design, if you're doing an audio only design, you should be aware that there's going to be tempo effects. Higher tempos will have higher effective empathy. But in general, there's no difference between fast and slow in heartbeats, except when there are no visuals, except when we're giving all of our attention to the audio. So, summary. When participants heard the heartbeat of another person, cognitive empathy changed. And 2, effective empathy increased. There were additional differences in empathic state due to the heartbeat tempo, the audiovisual congruency. Okay, let's go to the ECG results. This is the effect on the heart. So this is how I do heart rate measurement. So an incoming ECG signal looks something like this, where we have these peaks which represent the heartbeat. And so the first thing I do is I locate these peaks in the signal. Then I calculate the mean distance in between these peaks. And I do this over particular windows, 10-second window, 5-second hop. And then I subtract the baseline. So given a heartbeat stimulus, audio stimulus that's coming in like this, I would take the first 10 seconds before this as their quote unquote resting heart rate for the trial. So here's some results. Now, this work actually started off with piloting. It was almost all about the heart rate effects. And so I have some results here that show, first of all, that the heart rate shifts towards the tempo. Playing participants' fast heartbeats created a significantly higher heart rate than slow heartbeats. And furthermore, this is something that could be measured in less than 20 seconds. So hearing a heartbeat causes the listener's heart rate to shift. I did some other piloting that resembled the audio-only condition, but it was only the audio-only condition, no visuals. And I got this result. So this is a little bit different here, but it shows the effect of the heart rate over time. So we start off at 0, and then they change a little bit here. And this is the measured heart rate. The solid lines are the high self-reported effective empathy. The low lines are the low self-reported empathy. And there were significant effects. So there were heart rate differences between high and low empathy trials. So these solid lines were significantly higher than the low empathy trials. And then furthermore, when we went farther into the trial, we saw that there were significant differences due to tempo that seemed to only occur when there was high effective empathy. This is a really interesting result for us because it seemed to imply that empathy and tempo would also relate with each other. And so this was some of the motivating stuff that went behind this experiment. So let's look at differences in heart rate. So this is a comparison of the heart rate changes due to modality in this multimodal experiment. So this is a comparison of the visual only, the audio only, and the audio-visual condition. And this is the measured change in heart rate from baseline across all trials. And what this shows is a significant effect of the auditory heartbeat. In particular, the auditory heartbeat decreased the listener's heart rate. They became more relaxed, more calm, compared to the visual only condition, which seemed to have there's no significant difference between that and the baseline. So the visual only condition, when there was no heartbeats, the listener's heart was not affected. So the auditory heartbeat decreased listeners' heart rates. We also looked for differences due to empathy, like we found in piloting. And in this multimodal study, we actually found that specific thing that we had noticed was specific to the audio-only condition. So when participants were giving all of their attention to the audio stimulus, they had high heart rate, high effective empathy was associated with higher heart rates than low effective empathy. And this was specific to the audio-only condition. Let's look at heart rate changes due to congruency. So this was also a significant effect. It compares the audiovisual incongruent versus the audiovisual congruent, and there was a significant difference in heart rate between these two conditions. This is, again, a audiovisual comparison. But what this is showing is that audiovisual congruent is producing a significantly higher heart rate than the audiovisual incongruent. And so there was a significant difference in heart rate due to congruency. We've also looked for differences for differences in tempo. This is a comparison of the audio only and the audio visual conditions. And we did not find a difference due to tempo in this comparison. We were kind of wondering why this could be. And so there's a few differences between that and our piloting. One is that we have 4 times as many trials. So there's probably fatigue effects. Also, they're multimodal. So to explore the fatigue effects, we just looked at the first. So there was no significant differences in heart rate due to the heartbeat tempo. However, when we looked at the first 36 trials, this is the first, I guess, 18 minutes of the study, there were significant differences. So in particular, so the higher heart rates were associated with a relatively higher heart rate than the low auditory heartbeat. So this effect was present very early in the study, but then went away, likely due to fatigue. So here's a summary of the heart rate results. When participants heard the heartbeat of another person, their heart rate decreased. There were additional differences in heart rate due to empathy, audiovisual congruency, and heartbeat tempo. And these are all, there's more information in my thesis. All right, let's move to the EEG results. So to study to study the person's EEG response to their own heartbeat, I use something called the heartbeat evoked potential, which is measured with EEG. And you can kind of think about the physiology this way: that there are neurons that are going from the heart to the brain, and also that our attention is also constantly, we're constantly aware of our own heart. Okay, and so in order to look at the effects of attention to our own heart, we can look at epochs that are aligned to the R peak in the ECG signal. So you can imagine listening to the study. I'm measuring your heart rate, and then I'm going to make individual trials out of each heartbeat. And those are the data is actually what I'm recording in on your EEG. And this has been shown. There's been shown to be significant differences in this heartbeat evoked potential due to different conditions. And you can use this information to then project into the brain and see what parts of the brain might be involved. But in general, the HEP reflects cortical processing of the afferent internal cardiac signal. Any questions about that?
What does that mean?
So the HEP, it's The next slide will show more, but in general, the HEP is time locked to your own heartbeat. And so whatever, if there are differences in your own cortical processing between different conditions that are related to your own heart signal, those will pop up here. So this might help explain a little bit more. So people have conducted experiments where they ask people to count their own heartbeat in silence without taking their pulse. And people who are better at this have more positive HEP responses. Furthermore, when you actually distract somebody in the middle of one of these tasks, that has been shown to create a more negative response in this HEP. This has also been related to self-consciousness and the effective judgment of faces and emotional valence. And so in particular, what's interesting is that when people are doing effective judgment of other people, in this case, visual stimuli, this produces a more negative HEP. So it's like their cardiac attention has gone to another person. For that reason, we make this additional hypothesis, which is that listening to others' heartbeats will have a more negative frontal HEP, reflecting their attention shifting from themselves to the other person. So here's how I do my EEG analysis. It's kind of complicated, but first of all, you have to clean the EEG. This includes downsampling, high-pass filtering, removing line noise. We have to identify and remove any bad channels that just do not have a very good connection to the scalp. I use a tool called artifact subspace rejection to then clean individual channels, like little chunks of that data. Then we need to interpolate the removed channels. And then I re-reference to a common mode average. Then I decompose the EEG signal into components using independent component analysis. And so I run an independent component analysis on the entire experiment, the data from the entire experiment. And then after that, I epoch around the R peak. The R peak is their heartbeat. So I create a bunch of trials or epochs that are all aligned to that peak.
Epochs all equal length?
Mm-hmm. Yeah, they go from minus 200 milliseconds to 600 milliseconds.
So this is an evoked response we haven't worked with, so
Yeah.
I'm curious about it. Is there a latency? So this is in reaction to your own heartbeat. What's the latency of like P300 milliseconds? Is there a latency like that?
So it depends on the experiment and the task, but in the heartbeat attention one, it's between 200 to 250 milliseconds.
Okay, so it's a little faster.
So it's a little faster, but it's kind of all over the map. It's only been discovered since like 1985, but I think this has actually generated a lot of interest, specifically in recent years, because it has been shown to kind of reflect this social and other sorts of processing.
And from your mappings, are you seeing this over parietal? Because it
Yeah.
looks like you're seeing a little bit in left motor cortex.
Yeah, that's right.
Actually makes sense.
Yeah.
But,
It's very. It's.
is that mostly where you're seeing it?
Yeah. I'm seeing it in the. This will be on the next slide, but I'm seeing it in the frontal and the parietal.
Yeah.
So I'm getting two significant differences there. And people have actually used ECOG to try to localize even further, like where in the brain this might be happening. But it seems to be maybe the anterior cingulate cortex in the supplementary motor area.
Well, how do you actually differentiate that from just a regular P300 listening to someone else's heartbeat?
Well, so.
Is it because it's faster?
Well, the epochs are not aligned. I'm not doing a audio evoked potential, so I'm not aligning to the external heartbeat.
Yeah.
So I
But
guess.
if you're listening to someone else's, it is an auditory
Yeah,
stimulus.
it is.
So that's something to think about. Are you actually seeing a P3?
Yeah.
Or are you seeing an HEP?
Yeah.
That we should talk about.
Okay.
Those aren't aligned, so we're not epoching based on what so we wouldn't see.
Oh, okay.
Yeah. So it might wash out. Yeah.
Yeah. Okay.
But it is an interesting question. And then so once we have these components, we then model dipoles, which are not just like mathematical projections into the brain that could be creating these ICAs. And then finally, once we have all this data, once we've done this per participant, then we cluster those components to look at differences due to the conditions. I use EEG Lab study for this group level analysis. And the first thing I do is pre-compute a bunch of ICA features. So this includes the ERPs from the ICAs, the power spectrum, ERPs, related spectral perturbations, ITCs, and scalp maps. And I use all of these, I compute all of these. Then I cluster them by similarity. And so I'm using all the features to determine components that are similar to each other. And from this, we then look at components across participants. Now, with the HEP, one of the most important things to do is to remove the cardiac artifact, which is because we're time aligning to the R signal, we have the heartbeat actually in the EEG. And that is not an EEG signal, that is a heart signal, so we have to remove it. So ICA kind of makes this easy, or this clustering. So this right here is actually a heartbeat signal here. And there's other ones too. So this is another one. This one is a little bit less, but you can see that there's a prominent peak there, right there, and right there. Okay. So now these contain the cardiac artifact, and so they're actually just not analyzed. Now anything else besides that is true brain activity. And of these remaining components, a few of them seem to have differences in the time window of 200 to 600 milliseconds, which we had previously identified as being important for self-other processing.
You're using ICA to make the determination of what is and what is not a heartbeat?
Well, actually, the ICA actually takes similar components, and so some components like just have this big R signal, which has been decomposed or taken out of it, or separated from the rest of the signals. And so I identify this by I. That is because I know this is clearly a cardiac artifact. This looks like an ECG signal. This one too, and then the remainder are not as clear. But for example, this one right here. Okay.
Okay, I'm going to say that the bottom row, the second one along has a strong heartbeat component.
Yep, that's fun too. So, anyways, I didn't look at that one, but yeah, you're right. I would probably.
I made that up.
Yeah.
Right, so the second row, first one has a strong heartbeat. I'm asserting that.
Yeah.
By I. Yeah. They tell me why your assertions by I are better than mine, and why we should do.
Okay. So I think one of the important things to take into consideration. So I think we can both agree that these two are cardiac artifacts very clearly, and then I guess we're talking about these remainders, which are which just kind of which are maybe a little bit more arbitrary. I think one way to go about this is that I'm actually, in addition to this by eye analysis, I'm also looking for significant differences. And so it's not just that so the reason I'm not analyzing this one is because it looks like there's no difference between the visual only and audiovisual condition. Whereas this one, it looks like there is. And same thing with this one and that one.
So you're hypothesizing the difference between the visual only and the audio only. So you're only analyzing the data that are confirmatory.
Yeah.
That supporting your hypotheses.
Right.
That's essentially putting your thumb on the scale.
I could see how that would be.
Well, he's worried about artifacts.
Yeah. So this slide is really just about identifying artifacts and identifying the components to look for. So what I want to convey with this is that this particular method is very good at identifying, isolating the cardiac artifact, which in particular making components that are due to brain signal, due to not brain signal. It's very likely. I mean, I could totally see how every one of these components might have some sort of cardiac artifact in it. And I think that with my eye, if I was going to make a heuristic off of this, I would say that if there is a prominent if there's a prominent peak near zero that is above 0.1 microvolts, that I'm not going to analyze that. That I'm going to consider that as a heartbeat.
Another challenge to this is that all of these squares have different scales.
Yeah, they do. But that actually is reflective of the component ERP itself.
Yeah, so when I look at the bottom row, second one in, and I see a big peak, it's actually not a big peak.
Yeah.
If you compare it to the one right above it, because the scales are different.
That's true.
So that's right. So that suggests to me that to make that point that your decision by inspection is kind of valid. You probably want to see if you can put these on the same scale.
Yeah.
And,
Okay.
and a lot of the stuff will start to look like just jitter noise, and the spikes will.
Okay. That's a good idea.
Have you filtered for blink artifacts also?
Yeah, the subsequent ERPs, we do that. Yeah. And so from those two clusters, we look at the ERP and the cluster dipoles. And so this was the frontal cluster, and I'm using non-parametric statistics or permutation-based statistics to control for multiple comparisons. And so indeed, there was a significant difference between the visual only and the audio-visual condition. In particular, the visual only was significantly more positive than the audio-visual condition, which was significantly more negative. This localized to the anterior prefrontal cortex. Not much is known about this, but this is probably due to the task itself, which is a multimodal task where they're trying to complete a goal. And anyways, for this particular component, there was a significant difference. It's a frontal, more negative component associated with the audiovisual heartbeat.
And just to clarify, the nature of this task is blending internal and external. Right?
Yeah.
So you're trying to compare your heartbeat.
Well, so
No, you're not.
Yeah. So you're aware of your own heartbeat, maybe subconsciously, but your determination is based upon someone else's. Yeah. And so this other cluster, which we thought might be significant, also turned out to be significant, and that was localized to a more earlier, 200 milliseconds, and that was associated with a more positive ERP associated with the audiovisual condition, and a more negative ERP associated with the visual-only condition. And this localized to the superior parietal cortex, which probably reflects the multimodal nature of this, as well as the linguistic task of identifying the word associated with it. So the main results of this are that there were differences in the HEP between the visual-only and the audio-visual conditions. And particularly, the auditory heartbeat had a more negative frontal HEP, which, as we know from earlier, reflected a decrease in internal cardiac tension. So when participants heard the heartbeat of another person, their heartbeat evoked potential changed, and their cardiac cortical attention decreased. Let's get to the discussion. And I'm going to frame this entirely in terms of music. So, listening empathically to a sound that resembles the tactus of music decreased listeners' attention to their own heartbeat. This reflects an attention shift from the internal heartbeat to the external heartbeat. And this is a cortical mechanism that might underlie empathic listening to music. When we listen empathically to music, when we listen to it as if it's another person, or when we engage with it, our attention to our own heartbeat might be decreasing. Okay. Listening empathically to a sound that resembled the tactus of music decreased listeners' heart rate. Yes, Prue?
What is the tactus?
Tactus is the beat of music. So listening empathically to a sound that resembled the beat of music decreased listeners' heart rate. This might be related to a shift in the heartbeat attention. So the fact that our attention shifted to the other, our heartbeat attention is shifted to the other person, created a decrease in our heart. Maybe that has to do with this decrease in heart rate. But perhaps this is a physiological mechanism by which music relaxes people. So when we listen to music with our attention, as if it's another person, when we become more relaxed as of that might be the same effect we're seeing here. Those have to do with listening to music. I'm also going to present some other stuff related to music that might be more interesting for music research. Listening to a sound that resembled the tactus of music changed cognitive empathy. That is because the beat was perceived as affective information. So there was some emotional information, some emotional content in the beat. In music research, there's this notion of perceived emotions, which is our ability to perceive the emotions in a musical piece without feeling them. From my perspective, this has a lot to do with cognitive empathy. We're probably using very similar mechanism here. Finally, listening empathically to a sound that resembled the tactus of music increased affective empathy. The beat increased affective connection to the other person. Reed, did you have another question?
Yeah. It says change cognitive empathy during cognitive empathy, or are you just not making that kind of?
That's a really good question, but we're not doing that here. And so this is in music research, we also have this other idea, which is induced emotions in music. Our ability to feel what the music is actually conveying there. And to me, this has a lot to do with affective empathy. So a very similar mechanism might be used in creating this induced emotional effect in music. So concluding now, why is the heart a prominent lyrical metaphor for feeling? Because the heart is a locus of feeling, perceived and induced. Why does the beat of music resemble a heartbeat? To alter listeners' cardiac cortical attention. Finally, when do we feel with music? When we listen empathically to music, especially its beat. Okay, time for audience input. Can the heartbeat be used as an empathic technology? Yes or no? 1, 2, 3. Yay, okay. Thanks very much, that's it.